All right. Oh, let me pass this out. All right. Continuing on from last time. So, last time we talked about eukaryotic DNA replication. I'm going to go up a bit. Quick review. We talked about the different enzymes responsible for eukaryotic DNA replication. I guess Dr. Hasties gave you some extra information about DNA polymerase, where is it? Delta and epsilon. New information to me is the fact that DNA polymerase delta only replicates the lagging strand. I used to learn that DNA polymerase delta is the one in eukaryotes that makes DNA both on the leading and lagging strands. So, brand new information here. DNA polymerase delta, make sure you know, only replicates the lagging strand in eukaryotes. Meanwhile, DNA polymerase epsilon only replicates the leading strand in eukaryotes. You can contrast either of these to prokaryotes. What's the main polymerase in prokaryotes called? Polymerase 3. DNA polymerase Roman numeral 3 is the main polymerase in prokaryotes. Then there's another one called DNA polymerase 1, whose job is to finish replicating DNA on what strand in prokaryotes? The lagging strand. DNA polymerase 1 only replicates DNA on the lagging strand of prokaryotes. And it doesn't even replicate all the DNA on the lagging strand. Its only job is to fill in those gaps between Okazaki fragments on the lagging strand. Other stuff we did last time, we skipped these mechanisms because he never asks about the mechanisms. If he does this semester, or plans to, I will go over it during the exam review. But the other thing we talked about in eukaryotes is telomerase. The reason why this is an important enzyme or an important concept is that eukaryotic DNA is linear, not circular, and that kind of gives it a bit of a disadvantage on one aspect. Every time you replicate linear strands of DNA, they are unable to replicate the very last part of the lagging strand of DNA. So what ends up happening is that every time a cell replicates, if it has linear DNA, the DNA will get shorter every time that cell replicates. Unless that cell has an enzyme called telomerase. We learned last time that telomerase's job is to extend the template strand, the lagging strand template a little bit, by just bases that don't matter. It extends it with DNA. I might have said it extends it with RNA on accident. That is my bad if I said that. But it extends the template with DNA, and then DNA polymerase alpha will now read this new extended template to finish replicating the lagging strand of DNA. This prevents the shortening of telomeres in cells that have active telomerase. The reason why I keep stressing the word active telomerase is there's a common exam question that sometimes trips people up, where there's two versions of it. Either one, they ask, which of the following cells have telomerase? And the answer technically is all cells have the gene for telomerase. All cells have the gene for telomerase, but only cells that replicate often or replicate very quickly have active telomerase, okay? All cells have the gene for telomerase, but only cells that replicate very quickly and very often have active telomerase. The active ones would include stem cells, immune cells, germline cells, cancer cells. I think those are the examples that you're usually given. Those all replicate all the time. Other things we went over, for other cells not on that active list, they have a limited number of times they can replicate before they reach a limit called the Hayflick limit. This is the limit that is when these cells replicate, DNA gets shorter and shorter. The Hayflick limit is, well, the DNA has gotten so short that it has lost the ability to replicate again, or it contains a lot of mutations that it is unable to replicate anymore. Some information about telomerase itself. It is a reverse transcriptase, and we went over, I think that was the very last thing we went over last time, so let me jump down to the reverse transcriptase slides. Specifically, the one I am looking at is, I believe, this one. Nope, 1 second. Is this one. Reverse transcriptase is a special enzyme that bacteria have, that are in some eukaryotes, that have three activities here. Reverse transcriptase's main activity is this one right here, where it takes an RNA template and makes DNA out of it. That is what RNA-directed DNA polymerase means. Let me make sure this is still recording. Okay. Its second activity is this one, RNase hydrolase activity, whose job is to degrade the DNA-RNA hybrid that would have been made after step 1. Step 1 creates DNA out of RNA, so you have a hybrid here. The second activity breaks apart that hybrid. And the last activity is, well, now that you made brand new DNA out of that RNA, you can make more DNA from that brand new DNA that you just made. So DNA-directed DNA polymerase is you use your new DNA as a template to make more DNA. So a lot of viruses, a lot of RNA viruses have this enzyme packaged into their protein package when they infect cells. So that's how they're able to incorporate their RNA genome into a target eukaryote's genome. I gotta talk about cDNA next. Let me see if there's anything else. Oh yeah, you do not need to know all this mechanism for the exam. It even says that here. However, there is one statement on this slide that I think is technically stated elsewhere as well that you do need to know for the exam, and that's this thing right here. That reverse transcriptase is pretty tricky because it uses the host's own tRNA, any tRNA really, as a primer to do reverse transcription. So that is stated here. I think it is stated one other place somewhere. You do need to know that fact regarding reverse transcriptase. There's some other memorization facts that I never bother to remember each semester, but I will tell you that you do need to know for the exam, and that is, I think, really just these bullet point facts about RNA, about reverse transcriptase. Yep, that one we already went over. We already talked about the hybrid. We already talked about the RNA viruses. Oh, part of the reason why HIV is so hard to treat, and I believe those facts are kind of annoyingly scattered about in this section of the notes. One reason why HIV is so hard to treat is because it has reverse transcriptase, and when it incorporates its genome into a target cell's genome, it makes a lot of mistakes, which you'd think that would be disadvantageous for HIV, but really these mistakes are fine for HIV. What these mistakes end up doing is they create subtle differences in the enzymes that HIV is forcing the target cell to make for it. These small differences are just enough that if we design a drug to treat one version of HIV, that drug will be useless on the next version of HIV because HIV would have mutated already. Or even if we design siRNA targets, they're the same problem. If you have small little mutations that HIV has created, nothing that we make can target those mutations. So that's one of the reasons why HIV is so difficult to treat, is because its reverse transcriptase loves to make mistakes, and it is difficult for us to keep up with these mutations. Let's see, other things. Okay, yeah, cDNA. All right, so let me talk about that. Okay, so you might see here and in other classes a phrase called cDNA. This is different. This is not the same as regular DNA. The C stands for complementary. All right, and whenever you see the phrase cDNA, 99% of the time they usually use this in the context of eukaryotes, and here's why. If we were to create a database of some organism's genome, there's several ways we can do that. Meaning, if we want to know what are the genes that are in this organism, what genes are functional, which ones aren't, we usually first have to sequence the genome and store it in a database. If we straight up sequence an organism's genome, we create a DNA library. Okay? That is just straight up the genome of an organism. Or tissue or whatever, just it's the genome, the raw DNA genome. This means this DNA library includes genes with introns, exons, and even includes those spaces between genes called intergenic regions. A raw DNA library has all those things. It's straight up copy and paste to the entire genome of some organism. However, to contrast that, if you see something called a cDNA library, this is a library, technically, of all the types of RNA that an organism can make. And most of it is mRNA, so I'll put that in parentheses here. But it is a library of RNA that an organism can make. Why is it called cDNA then? So here's why. Let's say I want to trick a prokaryote into making insulin for me, right? Insulin is that, I use that example all the time because it's a little bit easier to understand. That we want to trick a prokaryote to make insulin for us so we can use that insulin for biomedical purposes. There come some problems with that. Why? Well, all right, so problem one. The insulin gene is eukaryotic, not prokaryotic. Problem two, and it kind of ties in with problem one, the insulin gene has introns in it. Do prokaryotic genes have introns in them? No. The answer is no. Prokaryotic genes do not have introns in them. Genes are packed very close to each other in prokaryotes, so this actually is a huge problem. So I can't just cut out the insulin gene out of a human genome. I cannot just cut out the insulin gene and just stick it into a prokaryote and then it'll work, because a prokaryote does not understand introns. It's going to transcribe those introns as if they're like any other gene, which is a problem. So what we found out we could do is, why not just take insulin mRNA, which, does anyone know, fully finished insulin mRNA, does it have any introns in it anymore? No. Insulin mRNA, mature mRNA, is just the exons. It's just in its completed mRNA form, ready to be translated into insulin. So there are no introns in this thing. It's only the exons. They take insulin mRNA and reverse transcribe it into what is called insulin cDNA, which is DNA that is complementary specifically to the mRNA. This cDNA doesn't have any introns, so it's different than the insulin gene that was straight up from the eukaryotic genome, right? This is special insulin DNA that doesn't have any introns. This thing can be inserted into a prokaryote. That's how we trick prokaryotes into synthesizing eukaryotic genes, is we take mRNA from eukaryotes, reverse transcribe it into cDNA, because that's the form of DNA that won't have any introns in it, then we insert it into a prokaryote. We do a couple other things, usually not really asked about, but I'll list them for general knowledge. A couple other modifications we do to this is we add a Shine-Delgarno sequence as well. Which we are going to talk about in more detail in chapter 30. The reason why we need to add a Shine-Delgarno sequence is, you'll learn in chapter 30 that this is a sequence that is found on prokaryotic mRNA that lets ribosomes know to bind to it. Okay, so eukaryotic mRNA and eukaryotic DNA genes do not have a Shine-Delgarno sequence, so we have to kind of add that in as well if we want to trick a prokaryote into synthesizing insulin. Again, main concept is this. I am briefly mentioning Shine-Delgarno sequence because that will be talked about in chapter 30. But for now, if you can't wrap your head around the Shine-Delgarno sequence thing, just stick with this main fact, is that we need to get rid of the introns out of insulin in order to trick a prokaryote into making it for us. So that's what cDNA is. Whenever you see cDNA anywhere else, it is mRNA that is reverse transcribed into DNA. All right. Let me just check to see if there's anything else I need to talk about with cDNA. Yeah, you can do this with any type of RNA, by the way. I mentioned mRNA as an example, but you can do this for miRNAs, siRNAs, and that's sort of what they talk about here. Let's see. All right, we'll talk about that. And retrotransposons. Okay. Two additional things in chapter, do they talk about CERT6? Yes, okay. So a couple additional things that sometimes show up on the exam, but these are like rare topics that sometimes show up, sometimes don't. And I will go, of course, in more detail about this during the exam review if they show up this semester. So I'm only going to point out what I have seen historically. One, retrotransposons. Retrotransposons are these weird genes in eukaryotes that regularly remove themselves from the genome and insert themselves somewhere else in the genome in eukaryotes. And no one really knows why exactly. They just observe that this happens. Again, retrotransposons are sequences of DNA in a eukaryote that, for whatever reason, remove themselves due to some enzymatic mechanism and move themselves somewhere else in the genome and reinsert themselves. Some of them even just copy themselves and reinsert themselves somewhere else. The one example of a retrotransposon that's talked about that I have seen on the exam is CERT6. There's some facts on here about CERT6. It's a longevity gene. You do need to know the facts, the biological functions of CERT6. I never remember them, but that usually sometimes shows up as a one question on the exam. CERT6 is which of the following? Deacetylase. Which, if you guys remember stuff about chromatin, what does an acetyl group do to chromatin? Remember?
It makes it looser, unwinds it?
Makes it easier to unwind DNA. So I'll jump back to deacetylase in a second. But when we talk about chromatin that has been acetylated, that we attach an acetyl group to, it means chromatin is a lot easier to open up at that spot, meaning whatever genes that happen to be there can be transcribed a lot easier than elsewhere in the genome. So, CERT6 is a deacetylase. It likes to remove acetyl groups, okay? You don't need to know more than that in regards to the deacetylase thing. Longevity gene, maintains telomere length. Like, look at all these facts about it. Please memorize them, and you know, it'll be like which of the following is true or which of the following is false about CERT6 on the exam. So I'm not going to go over to detail because it's memorization. They give you even more detail about it. Really, you're not going to get a million questions on CERT6. You're going to get 1 or 0. So feel free to look at that. More interesting facts about retrotransposons. Feel free to look at this. But again, it is a not often talked about topic on the exam, so I will not spend time on it either. Let me see if I missed anything. Oh, double-strand break repair. This is more discussed in detail in like a genetics course, but basically sometimes during DNA replication, during cellular replication, DNA replication, mitosis, or meiosis, this happens in either one, but is more critical in meiosis. Chromosomes have to come together in order for you to sort your genes, in order to create genetic diversity, chromosomes, homologous chromosomes come together and sometimes switch loci. That creates genetic diversity, and usually everything's fine. However, sometimes, and I don't know if this illustrates it here, it does not, sometimes during this switch, chromosomes can stick together, or sometimes one chromosome gets a little bit more DNA than they're supposed to, or sometimes both chromosomes lose DNA, not even switching loci with each other. They sometimes both completely delete large chunks of DNA. When that part happens, there's usually such a big issue that the normal DNA repair enzymes are unable to fix these. In fact, we only have a few enzymes that are involved in repairing this double-strand break repair thing. You may have heard of two of them before, BRCA1 and BRCA2. Does anyone know what disease kind of involves a mutation in these genes?
Cancer?
Specifically breast cancer, yes. Breast cancer, BRCA1 and BRCA2. Usually breast cancer is associated with a mutation in one or both of these things because their job is to repair double-strand breaks. And really, if for the exam, the only thing I really notice when they talk about this is, you know, which genes repair double-strand breaks. It's really listed in red right here, BRCA1 and BRCA2. I'm really just drawing at straws here. These things are not usually asked about. I'm just going by what I have seen in the past. And that might be it for 28. I was surprised how quickly we went through 28. Yeah, that's it for 28. So, review. I'm not going to go over Messel's install. It takes a million years. Okay. In the diagram below, label the leading strands and lagging strands at each of the replication forks. So imagine, you know, top left, top right, bottom left, and bottom right. This is very similar to a question you can get on the exam. They will give this, and they will have letter A up here, B there, C there, and D there. And it may ask which things on here are the leading strands and which ones are the lagging strands. So, anyone remember how to figure out leading and lagging strand? Let's see. How about someone want to take a stab at this? Someone tell me which corners here, top left, top right, bottom left, and bottom right, will be your leading strand on DNA. You can take time to solve it. You need to draw it out and everything. Go ahead. Try to figure it out. Which ones are your leading strands on here? I have to do it too. Let's see. We split this. My tip for you here is split this in half and then draw out where the new template DNA would go and how it's made. Yes, you have an idea?
So the bottom right is going to be leading.
All right, let's see. Bottom right is written 503' as the template. So that means your new DNA is going to be generated in what direction? Going to the right or going to the left on this picture? It'll be 3' to 5' on the bottom, which is to the right on the bottom strand, right? So, if I won't be able to draw it on a Word doc, but new DNA on the bottom right is going to be made going to the right, which is the same direction that the replication fork is going in on the right half here. So, replication fork is going to the right, new DNA is going to the right. Therefore, that is your leading strand by definition, because the direction that new DNA is being made is the same direction as the replication fork. So bottom right is a leading strand. That gives you a shortcut, right? That means the top left is what? The top left, so I'm kind of cheating a bit. The top left is also leading. Whenever you can find one corner, okay? If the bottom right is leading strand, immediately write down the diagonal opposite, meaning if bottom right is leading, that means top left is also the leading strand. Okay? The remaining two, therefore, have to be lagging strand, right? So, write it in whatever order you want. Bottom right is leading strand, top left is leading strand. If the question asks which ones are lagging strands, well, it's the two remaining ones we haven't talked about. The top right is a lagging strand, and the bottom left is a lagging strand. Feel free to review the SI sessions from earlier in the week to see how to draw those out. Okay, but if you do this enough, you can sometimes identify without having to draw anything or doing a very quick illustration. All right, let's see. I'm not going to answer every single one of these. Yeah, some of these also are more specific than what you need. Activities, that's very important. We'll do DNA polymerase 3 first, though. DNA polymerase 3, is that one subunit or many subunits? Many subunits, right? I think it's like 4 subunits, right?
Alpha
Alpha.
says 10.
Oh, there's more probably. We learned about how many in DNA polymerase 3. Alpha, beta, epsilon, right? And theta, I believe. Oh yeah, you guys notice how confusing this is? Because DNA polymerase 3 is a prokaryotic polymerase, but it is made of subunits with Greek letters in them. What about eukaryotic DNA polymerases? How are they named? With Greek letters in them. Over and over again, I stress that you guys need to split up your notes into different sections for prokaryote and eukaryote so you can hold them next to each other and compare them to each other so you don't get these confused. I guarantee people will confuse the Greek letter subunits of this prokaryote polymerase with the Greek letter polymerases in eukaryotes. So, with that warning out of the way, DNA polymerase 3 subunit alpha has a particular activity. Anyone remember what it's called?
Yeah. 5' polymerase.
5' to 3' polymerase, which basically means what does this subunit do?
Replication.
It's the one that replicates DNA, exactly. This is the main one. Okay. Beta, there's another nickname for the beta subunit. What is it? The sliding clamp, exactly. The sliding clamp's job is it gives DNA polymerase 3 enormous processivity. Good. Basically, Basically what that means is what this means is DNA polymerase 3 is able to replicate tons of DNA without stopping. Oops, misspelled this. Epsilon, what activity does the epsilon subunit have?
3' exonuclease.
Exactly, 3' exonuclease, which is sometimes also written as 3' to 5' exonuclease, so don't panic if you see answer choices written like this instead. It means the same thing. This is your editor function with DNA polymerase 3, or any polymerase that has that activity. What this activity's job is every time the wrong base is added during DNA replication, this activity will remove that incorrectly paired base so that DNA polymerase 3 can put in the correct one, to try again and put in the correct one. This is your editor though, which activity actually detects the error, proofreads the error in the first place.
Alpha.
It is the alpha subunit, exactly. This is also the proofreader because and it's annoying because in other classes you might learn that this is your proofreader, so I have to tell you that for this class, the 5' and 3' polymerase is your proofreader because what this activity is doing, it's the one adding nucleotides over and over and over again. It's going to add sometimes the wrong nucleotide and try to fit together the wrong puzzle piece with the wrong place. And it knows it would make a mistake in that moment. So it is proofreading itself. Then, however, it doesn't have the ability to remove the incorrectly added nucleotide. So it has to communicate with the epsilon subunit, which will actually remove the incorrectly added nucleotide. So that's why epsilon is the editor. Theta, we don't know what it does. All right, and that's it for DNA polymerase 3. Oh yeah, it is also your main polymerase, right? It's the one that makes most of your DNA in prokaryotes. DNA polymerase 1 is different. Is DNA polymerase 1 one subunit or multi-subunit?
One subunit.
It is just one subunit, exactly. However, confusingly, this one subunit has 3 activities, all on a single subunit. That is because this enzyme has 3 active sites. Okay.
We're using the word domain and activity synonymously, right?
Domain,
Because I see sometimes we use like differently, but they seem kind of like the same thing.
You in actual in the lab and in probably in genetics because they more accurately use these terms. Yes, that domains each define a particular function or characteristic of a particular protein, such as a transmembrane domain. There's a particular pattern that if someone finds this pattern in this brand new protein, they know it's going to go and stick itself in the membrane. or a domain to replicate DNA. That would be domain versus activity. It usually means the same thing. I don't know if in this class Dr. Hasties is consistent enough to be using that.
So we see it, what do we like?
See if you
on-
see
on
the
the
phrase
test?
domain. Nah, if you see domain, I don't think I've ever seen them use the phrase. I think maybe in chapter 29, the one time he uses the phrase domain is to describe transcription factors, because transcription factors have what is called a DNA binding domain, meaning that any novel protein, if it has a DNA binding domain, it is a transcription factor. I think that's the one time I see him use the phrase domain, at least on the test. If I run into another one that I had forgotten, I'll point it out then also.
Okay.
So, 3 active sites, 3 activities. 2 of the activities are the same as in DNA polymerase 3, 5' to 3' polymerase, meaning DNA polymerase 1 makes DNA. It also has 3' exonuclease, meaning it also has the ability to edit DNA. However, the 3rd activity is unique just to DNA polymerase 3, and that is called 5' exonuclease. Okay? This one, more generally speaking, removes chunks of DNA ahead of the enzyme on the site that it is on. Specifically, we learned that 5' exonuclease removes primers and replaces, well, it removes the primers so that the rest of the enzyme can replace it with actual DNA. Because primers are made of RNA, we need to remove those and replace them with actual DNA. So that's what 5' exonuclease does, and that only DNA polymerase 1 has that ability. So, which strand are you going to find tons of DNA polymerase 1 on, the leading or lagging strand? The lagging strand. Because the lagging strand, every single Okazaki fragment has a primer attached to it. So, DNA polymerase 1 is going to be heavily involved on the lagging strand. So, going back to this picture up here, this could be rephrased into which area on here, A, B, C, or D, has DNA polymerase 1 on it. Just pick the lagging strand. It's another way of asking you which one is the lagging strand. Oh yeah, the large cleanout fragment. So, we reviewed that a bit. Basically, the large cleanout fragment is modified DNA polymerase 1. I'm not going to go over it in detail, but it's modified DNA polymerase 1. The large cleanout fragment is polymerase 1 with just those 2 activities. That's it. Basically, the large cleanout fragment was made by cutting off the 3rd activity, so that all you have left are these 2. Therefore, you can use the large cleanout fragment to synthesize DNA in the lab. Let's see, other review things. We already talked about DNA polymerase 3. Oh, I didn't know I included this. That's great. So yeah, where would you find DNA polymerase 1? You can practice that. Make sure you're able to define and understand what each of these terms mean. Some of them are easier than others. Bidirectional replication is pretty easy. This means that for every origin of replication, you create 2 replication forks that each replicate DNA in opposing directions. Make sure you review what each of those do. Nick translation we talked about. Nick translation is that process of DNA replication that only occurs on the lagging strand, and it involves not only DNA polymerase 1, but what other enzyme is necessary for Nick translation? Ligase, exactly. Ligase's job is to connect the sugar phosphate backbone between Okazaki fragments on the lagging strand. Prokaryotic chain termination, meaning how does DNA replication end in prokaryotes? Remember that DNA is circular in prokaryotes, however, you do need to communicate to the replication forks to stop replicating DNA. There's a particular site on prokaryotic DNA that is necessary for this. Anyone remember what the name of that site is? The ter locus, which stands for the termination locus, the stop here locus. And there's an enzyme that is necessary, a protein that binds to that. What is the name of that protein? The TUS protein, exactly. You can, for those listening on the video, you can all review all of that in your notes. The TUS protein has what activity to help stop replication? Contra helicase. It stops the helicases. Good. You do need to know the different DNA polymerases in eukaryotes, all those Greek letters, alpha, beta, gamma, delta, and epsilon, and what each one does. So please review that. And pCNA is the eukaryotic version of sliding clamps in prokaryotes. pCNA is in eukaryotes and is the eukaryotic version of the sliding clamp in prokaryotes. We talked about the Hayflick limit and telomeres. We talked about reverse transcriptase. The phrase homologous chromosomes. I probably forgot to define what homologous chromosomes mean. Homologous, remember how we have 2 copies of every chromosome in eukaryotes? The homologous chromosomes are each copy of chromosome that is exactly the same as the other one. We talked about single-stranded break repair, double-stranded break repair. We talked about retrotransposons. That's about everything I could think of in chapter 28. By the time the exam review comes about, and if there's more unique things on there, I will talk about it during the exam review, but those are the core things for chapter 28. All right, enough stalling. Let me go into chapter 29 a little bit, since we're running out of time. And we are way ahead of time, I believe, right? The exam isn't until the 15th. Let's see, how many more SI sessions do we have? 1, 2, 3, 4, 5, 6. And I think I'm gonna try to have the exam review either Tuesday, Wednesday, or Thursday, either the 9th, 10th, or 11th. I have to see what day there's a big enough room that we can use. And then your exam is on the 15th. Okay. So, chapter 29. 29 is in 2 parts, 29A and 29B, and this talks about transcription, which in my opinion is the most important thing of the central dogma, is transcription. You have the greatest variety in terms of transcriptional regulation, in terms of regulation of gene expression, you will find at the level of transcription. There's certain things that make it easier to transcribe genes, certain things that make it harder to transcribe genes. At the DNA level, at the RNA polymerase level, which you'll see what that is in a little bit. But this, I think, is the most important component of the central dogma. 29A talks about prokaryotic transcription, and 29B talks about eukaryotic transcription. So, to begin with, you don't have to memorize names of theories or postulates or anything, but this is a good introduction to what mRNA is. Before they had even characterized it, what mRNA is, they knew had to be something that linked DNA to protein. Why? Because we knew what DNA was, and we knew that proteins are the functional component of cells, so there needed to be a medium between the DNA and the protein, and that is mRNA. They figured out a couple properties of them, which are listed here. Oh yeah. High turnover rate, for those who don't know what that means that it is constantly made and degraded. So mRNA, that is true for mRNA, it is constantly made and degraded. Here's a review of the central dogma of molecular biology. DNA replicates itself, DNA can be transcribed into RNA, and mRNA can be translated into protein. Nice quick review. Other forms of RNA, we were introduced to this a while ago, I think in chapter 10B or 10A, so you can review that in more detail there. Let's see. Oh, interesting fact here. The mechanism of transcription, which is to read DNA to make some type of RNA, is actually quite limited. Of your entire genome, of all the genes that you have, of several tens of thousands of genes that you have, only 1% of the time are being transcribed. Most of the genome is silent, depending on what cell or tissue that you are looking at. And this is most of the time. I actually think, and it's not listed here, but just for contrast comparison's sake, when an organism is in the development stage, meaning after fertilization and when the cells are replicating and you're growing complicated tissues in the development stage, transcription is at its maximum during that because you have to make a variety of tissues during development. So transcription's at a maximum. I don't know what percentage it is, but as a comparison, it is definitely going to be way more than 1%. This 1% fact is for regular cells and tissues. All right. Okay, sense and antisense. This is sometimes confusing. I will briefly talk about it now. Let me write an example real quick. make sure I do this in the correct directions. T5'. Okay. So, the phrase is sense and antisense. I'm going to label some stuff first. This is template. This is non-template. Okay. So, the way transcription works is an enzyme called RNA polymerase is going to read DNA to make mRNA. It will only read one strand of DNA to do this. It only needs to read one strand of DNA to do this. So the one that it happens to be reading is called the template strand. So RNA polymerase is going to read this to create mRNA. So let's make the mRNA strand. Just like with DNA replication, RNA polymerase will read the template in the 3' to 5' direction because it's making mRNA, new mRNA, in a 5' to 3' direction. So it'll read a T here and put an A down. It'll read an A there and put a U down. And that's a G, and that's a C, and that's a C. And this is another A, 3'. This would be the mRNA that will be made by the RNA polymerase. This is your mRNA. Okay? There are phrases that are sometimes used to describe DNA, such as the different strands of DNA, such as sense strand and antisense strand. Here's my definition of it. It may not be the exact textbook definition, but it'll very easily help you identify which is which. And that is your template strand is your antisense strand. And your non-template strand is your sense strand. Okay? So when we use phrases sense and antisense, we're talking about transcription because we need to know which one is the strand of DNA that is being read to be transcribed and which one is being left alone. The template strand, it sounds backwards, but here's the reason why. The template strand is your antisense strand, and the non-template strand is your sense strand. Why would the non-template strand be the sense strand? I will point your attention to this comparison here. Compare your sense strand of DNA to your mRNA, and what do you notice?
They're both 5' to 3'.
Both 5' to 3', and take a look at the bases as well. They're basically the same. The only difference are T's are now U's. That's it. So the reason why we call the non-template the sense strand is because this is basically the mRNA, what the mRNA will look like. Looks like this, except T's are now U's. That's it. All right, that's why we call it the sense strand, is because it actually will look like what the mRNA strand is. Is that it? All right. Good timing, because we just finished that.
That's it, class is over.
All right, so we'll continue this on Monday. Whoever has a sign-in sheet, if you could give that to me. And I'll see you guys next time.